Welcome to another episode of Progress, Potential, and Possibilities, discussions with fascinating people designing a better tomorrow for all of us. I'm your host, Ira Pastor. Welcome, everybody, again to another episode of our show. Today, bringing another really awesome guest involved in some really cool areas of science and technology that are creating a better tomorrow for all of us. Today, we are headed back to our friends at the Institute for Systems Biology, and I'd be honored today of being joined by Dr. Sui Huang, who is a professor at ISB. His lab is focused on studying how gene regulatory networks ultimately control gene activity to create these stable cellular states, different cell types, and ultimately how these state transitions lead into different lineages in both healthy and disease conditions. Dr. Wong is a molecular and cell biologist. He has a strong background in theoretical biology and has devoted much of his career to understanding the phenomenon of cancer from looking at it from a complex systems perspective. Before he joined ISB, Dr. Wong held faculty positions at University of Calgary at the Institute of Biocomplexity and Informatics. There he helped establish biocomplexity as a specific discipline in both research and teaching. He was also at Harvard Medical School Children's Hospital. There he first obtained experimental evidence for the existence of these high-dimensional attractors and mammalian gene regulatory networks. Dr. Wong grew up in Geneva and Zurich. He did his medical degree from University of Zurich and then went on to do a PhD in their program for physicians, focused on molecular biology and physical chemistry, looking at interferon, and then went on to do a postdoc at Children's Hospital in Boston, investigating tumor angiogenesis and cell growth control. He also in that period studied in dynamical systems within affiliation through the New England Complex Systems Institute. A lot to get into today. Really excited about this episode. Dr. Sui Huang, thank you so much for taking time to come on the show.
Yes, thank you very much, Ira. Very good pleasure to be here.
Yeah, it's really great having you. I've looked for this episode now for a few years, and I'm glad we had the opportunity to set it up. So I'd love to start off, if we could, going back about 20 years now to your dissertation. I know I mentioned in your bio that you focused on interferon during your dissertation. After that, you went on to Donald Ingber's group to do a postdoc in angiogenesis. And, it's interesting because both of these areas, in their respective times sort of had a holy grail aura to them. But what we found out sort of slightly afterwards was, yes, they were great targets, but ultimately they had sort of double-edged sword components to them. Interferon, of course, too much, not good, too little, not good. And angiogenesis, it works, but there's also, things it does to the tissue microenvironment that aren't too good. Take us back about 20 years to when you were doing your dissertation and your postdoc, and a little bit is what you say on your website, how this experience, as you say, was a humbling recognition of profound complexity and fostered your desire to move beyond the reductionist, deterministic thinking that we saw in biology at the time.
Yeah, thank you. It's so funny because the way you summarized what my perspective evolved, it's just an emergent property, like a complex system is exactly right. So I never done that before, this long arc of development. Well, so I started after medical school, and you want to do something more fundamental and contribute to disease. So I chose what is super hardcore, cool science, which is at that time, our ability to manipulate the genome in more complex organisms. It's not CRISPR, didn't do much new other than accelerate the process. But at that time, we just started to be able to specifically target a gene as opposed to put in a gene in the organism. And so I had the great opportunity to work on a knockout, one of the first knockout of an immune system gene, this interferon-gamma, around XN people, Michel Aguet, Charles Weissman, Rolf Zinkenagel, and I just learned a lot, because you learn that what you read in the textbook, what genes do, the functions of a gene cannot just be defined as that. If you take it out, you see all possible things. I thought interferon-gamma was already known to be very much involved in cancer. That was known because in vitro experiments, you add interferon-gamma and you reduce cancer growth and so. But when you knock it out in the organism, it just totally unexpected things happen. You cannot tie it to the notion of causation of a disease. So we tried many inflammatory disease, rheumatoid arthritis, EAE, the encephalitis, allergic encephalitis, which is a model for MS or multiple sclerosis, thyroiditis. It's just not conclusive, right? Depending on how long you look at, what time scale you have, you can come at the conclusion that interferon-gamma is either promoting a disease or mitigating a disease. It's just not clear. Notion was that I put in all kinds of nasty oncogenes into these interferon deficient mice, couldn't get them to have more tumors. I wanted to show that the immune surveillance actually is real. And it was against my advisors. Rolf Zinkenagel, got a Nobel Prize for MHC. He said, no way, the immune system is important because, and that's correct, immune deficient people and people with immune, who lack T cells and B cells, they don't have more of these epithelial cancers that affect the elderly. And we also know the Down syndrome people who have totally messed up dysfunctional B and T cells, they have much, much less cancer. So something was wrong. And then it occurred to me, we just need to be more quantitative, and learn the details. And at that time, complex systems idea and nonlinear dynamics was ascending as a science to be taken seriously. Then 1993 came out a book, some books on the Santa Fe Institute and the book of Stuart Kaufman, Origin of Order. And I read that and I start to understand that you need to understand, to learn nonlinear dynamics. I think it's a core foundational science for understanding living systems. But that is still low dimensional. And then came all the omic sciences, and then which means that we have to do nonlinear dynamics in high dimensional systems and learn how that then lead to the emergence of phenotype and things we see, disease, health, that are again low dimensional. So that's roughly how I got to where I am now, and it's getting more exciting every day.
Absolutely. And so there's, I want to spend a little time introducing to our audience a series of concepts that I think is very important for them to understand. So when we get to sort of the big picture or what you're doing now, it'll make a lot more sense. And, the first of these that I'd like to spend a little time on, and I realize you probably devote entire semesters to these topics, so I appreciate we'll distill them down, is the whole principle of an attractor state, specifically in a biologic system. So we have sort of this area defined as a set of conditions that ultimately leads to this stable point. We don't know if that stable point is the development of an organ or tissue morphogenesis or wound healing, whatever the case may be. And as you've put in your papers in the past, sort of how cells ultimately sense this higher level need, right? The cardiomyocyte in my heart doesn't know it's going to make a heart, but there's something else there besides just the genes and proteins, and how ultimately that sort of hierarchical structure occurs. And I'd love to spend a little time on this because I know this is, you've been publishing on this theme for a couple decades now, but introduce us to the principle of biologic attractors, if you would.
Yeah, glad to do that. Thank you for, again, summarizing the big picture. I think we have to simplify something, and it's fine to simplify things, right? One problem when we try to explain this concept is that people always criticize you of leaving this and that details, their favorite details. But I think in biology, we need to learn or to acquire the courage to simplify things, because some details get in the way of seeing the big picture, but we have to simplify in a clever way so that we can then reverse the simplification later on. So for example, Galileo, you just forget about friction, and then that lead to discovery of the laws of motion, and then engineers can later add back friction, right? So the equivalent in biology would be, let's take the 20,000 genes, and we know genes command a phenotype. And the simple idea coming back to Mendel is a gene maps to a phenotype. You all know textbook one-to-one mapping between gene and phenotype. And but if you have 20,000 genes, would mean with 20,000 phenotypes, which probably is wrong. We all know that. So they collaborate, interact to produce more what we call complex phenotypes. And what is this process? So if these 20,000 genes wouldn't be interacting, then you can imagine a space of configurations where any combination of the activity status of these 20,000 genes would be a phenotype. That's already a notion of a combinatorial thing. But since the genes don't interact, we would have, if every gene is on and off, 2 to the 20,000, a huge space, a continuous space of phenotype. And the gene interact, like neurons interact, people interact. Whenever there's a complex system of a set of components and the components interact, then you actually need relatively few or sparse interactions to dramatically change the space of configurations that are possible. For example, if gene A inhibits gene B, then all configurations where A is high and B is highly expressed or active would be logically prohibited. And so you can imagine all kinds of combinations of genes talking to each other, either activating or inactivating each other. And that leads to a dramatic reduction of states defined as the activity state of these 20,000 genes that are likely to happen. And one can mathematically treat that for all kinds of interaction, network architectures, and then find that a relatively sparsely connected network would dramatically reduce that indifferent space of possible configurations of gene activities into few ones that are very likely, some that are less likely, some are impossible. Now, if you would use that quantity of how likely a particular configuration of these 20,000 genes can exist, let's say as a color in the space of all possible configurations, then you see patterns emerge. And that's essentially what we now learn, right, in the world of neural network and AI, that you have latent spaces of possible configurations. And that latent space is actually itself a characteristic topography, a landscape, in that now we can say, there's more probable states are kind of in valleys, and the other one that less probable can be depicted as in hilltops. So that structureless continuous space of all possible configurations of gene activities among these 20,000 genes will kind of fall up into a landscape or some complicated structure which has a lower dimensionality. Some people call this the intrinsic dimensionality of a set of configurations. And that landscape, the structure is determined by the nature of these interactions between the genes, which is fixed. That's our gene regulatory network. So that's a rough idea. Now, being more specific, then in that landscape, you have this, if the elevation of a landscape that we figure out, we are imaging now this structure of this, the structure of this configuration space is as a landscape, and we can imagine that the higher elevation states are the improbable states, configurations that violates more of these interactions. Right? If A inhibits B, then if A and B are high, every state which has that configuration of A and B would be less likely, therefore higher elevation. And then you would have states that are highly nicely coherent in terms of satisfying all the interactions, and there will be the lowest points in the landscape, the potentials, the lowest potential wells, so-called attractor states. So the natural configurations that the whole system would settle in if all the interaction prescriptions are satisfied. So these are the attractor states. And that's a very generic description. Many people have articulated something like that in different ways. Stuart Kaufman first to propose that for higher dimensional system with many, many genes. But before that, we have Max Delbruck. The people who actually discovered that genes regulate each other, Jacob and Monod, all propose that these interactions will reduce the space of possible configurations of activities to a relatively smaller number of stable states, the attractor states. And they are by nature of the topology of these landscapes separated by unstable states. And that leads to this discretization of more complex phenotypes that we see, right? 5-finger, 6-finger, skin color, light and dark. So all the discretization of phenotypes come from interactions. Otherwise, we would have a, undifferentiated continuum. So we have this landscape now. And then what can we do with that in terms of medicine? So that's very quickly, maybe you will ask that. With that picture in mind, we can imagine that every pathological state is essentially already preempted by the interaction of the genes in that gene regulatory network that is hardwired, because every gene interacts with others according to the structure of the proteins that is in the genome sequence. So we have this complicated landscape, and some states are not desired, and others are the ones that we see that are desired, that map into gene expression configurations that make sense. And that's the broadest picture. Of course, I think many people know, Stuart Kaufman first proposed that these natural coherent interaction configurations, due to the interactions, the attractor state will correspond to cell types as a phenotype, right? Every cell type has a particular gene expression configuration that is quite universal given a species, because we have similar gene regulatory networks, very similar ones. And so we have natural emergence of gene expression configurations that nature can then use as cell types. And evolution is essentially nothing but rewiring that gene regulatory network such that this landscape makes sense for cell types to move from one attractor to another and have a development towards a repertoire of cell types that makes sense in the adult. So that's, in a nutshell, the entire idea, from which naturally you can derive what pathological states are.
Right. And you actually introduced the second of the themes in your description there, and I just want to stress this for the audience. So we have our range of possible attractor states, which represent different cell types. If we hop out of those different states, we could make a normal cell cancerous or whatever. So there's other possibilities than just the regular cell lineages, but there's not infinite possibilities there. And you mentioned the term landscape, and this is just a theme that I just want to throw in here, because when we get to sort of some of the therapeutic concepts that you developed, I think this is important, because you've also written quite a bit on sort of this misunderstanding, let's say, of the term epigenetics. The landscape you're referring to, you refer to sort of that of Conrad Waddington, an epigenetic landscape. Not epigenetics, as most people that have listened to the show think of in terms of DNA methylation, histone modification, and all that, the molecular epigenetics. But we're talking systems epigenetics here. And I think this is an important point, because everything you just described in that landscape and all the underlying gene regulatory networks and everything else is happening is epigenetics in the traditional sense. And just say a couple words about this. I think this is important, and we'll link to some of your papers, like towards the unification of the two meanings of epigenetics in 2022.
Absolutely. You highlight a very important point. There's a lot of misunderstanding based on terminological inaccuracy, to paraphrase, some people. Uh, it's just, it's sad. It's just a semantic misunderstanding. So term epigenetics was, as you said, of course, coined by Waddington, actual previous people, epigenesis and then epigenetics.
Yep.
For these complicated interaction causal networks, as they call it, then they lead to a more higher level phenotypes, not necessarily in the genes. And in the 1970s, when people, I think it was Holiday, discovered DNA methylation, they just needed a term and they just call it epigenetics, probably with some loose allusion to Waddington. But that meant something else, totally different, right? So these kind of misunderstandings happens a lot where the same term means two different things.
Yep.
We know that is the basis of many jokes, like if people say, would you like some chicken pot pie? And then somebody say, yes, of course, I like all three of them. So if you take something out of context and things have different meanings, then the individual meanings get their own life. And so epigenetics then got hijacked, I think it's fair to say that, by molecular biologists to mean modifications of DNA that is covalent. At that time, quite stunning that something covalent can happen and without altering the genomic sequence. Just quickly on the story of that, I think it's important. And in the 90s, I think there was a roadmap on epigenetics. Already many good biologists sense that will lead to something confusion, so they opposed that roadmap, NIH roadmap. And there was a famous letter many people co-signed in Science. It's more a marketing term, epigenetics, where we use it in terms of now we have single cell epigenetics and looking at DNA methylation and fragmentation of chromatin-free regions. It's more a term that we have to be careful because it's just more essentially covalent modifications, right? So imagine the protein people would have used this similar marketing gimmick, and they would call all covalent post-translational covalent modifications, they would call it epiproteomics. Right. Then we would have a new field arising called epiproteomics. We would have NIH programs called epiproteomics, but we wouldn't have that because they didn't have this, marketing sense to name something. So that's epigenetics today, and that's totally different from what Waddington and other people meant by epigenetics. So sorry, it's a bit long, wandering discussion on the term, but I think we can also, they're related in some way, right? Because these DNA methylations affect gene expression and gene expression configurations across the genome is essentially what defines attractor states and your position in this landscape. So in some way, they are related. But as you know, DNA epigenetic modifiers, they are not locus aware, at least in mammalian, in humans. People have been looking for whether DNA methylation can sense some genome sequence and specifically methylate individual genes. Because you need interactions between gene loci to generate gene expression patterns that then define the landscape. Right. And so all these chromatin modifier, like polycomb, and so not in Drosophila, but in humans, they don't seem to have sequence preference. And so, which means all these epigenetic modifier that writes these epigenetic marks cannot contribute to gene expression patterns. So the reality is that these enzymes are recruited by transcription factors that can recognize specific sequences. They're almost all of them are recruited by transcription factors, and that is what affords a gene expression configuration, a particular character of how they comply with gene regulatory interactions, yes or not, and that leads to the landscape. So you need interactions between the gene loci of the genes that is not provided by the so-called molecular epigenetic apparatus, or this DNA methylating enzymes and demethylating enzymes and chromatin modifiers. You need transcription factors. Those are the engine of the gene regulatory network dynamics because they mediate the interactions between the genes.
Right. Excellent.
So,
no, perfect.
On a mundane problem actually of semantics.
No. I want to lay this out because it's going a very important place, and now sort of the third leg on this stool. Now we're going to bring cancer into this. You've written extensively the last couple decades on the non-genetic basis for cancer progression and metastasis, and again, we've had this somatic mutation model for how many decades now, and as you pointed out, there's so many things where it falls apart. You point out that although we've discovered hundreds of genes over the years involved in cancer formation, the transcriptomic heat maps of different tumors kind of line up. I mean, there's not an infinite array of transcriptomes. You talk about sort of foreign body tumor formation, spontaneous remission. These things don't explain mutations. And then most importantly, and I'll take my I'll give a cheer for Philadelphia because I'm sitting here, but Fox Chase Cancer Center is right over here, and this is where Beatrice Mintz back in 1974 did her seminal work on tumor reversion in teratocarcinoma, and there's an upgene. I don't know how many papers now that show, you take a normal cell, you put it in a different environment, it becomes cancerous and vice versa. Ultimately, what this all boils down to is that we need to think a little differently. And so I'd love to, we'll get to sort of the Sui Huang vision of cancer and therapeutics in a minute, but I want you to bring this in because it's extremely important in the principles that we just talked about in terms of attractor states and epigenetic landscapes, that there's much more going on than just the mutation. So please.
Yes, first, you accuse me of doing something that I haven't done by myself, but with a lot of complices and help, right? And that is pushing back. Many people are aware of that, implicit or explicit, but still the default notion is genes and their alterations are needed to explain an alteration of physiology toward malignancy. That's still there. But, when we write those things, I'm aware that people are aware, just don't act accordingly, right? It's like Jutta Folkman told us, right? The 4 stages of mourning, and it's also 4 stages of, responding to paradigm shifts, right? Anger, denial, then acceptance. And then what you see in science a lot is we knew it all along. And this we knew it all along syndrome is everywhere, but we still need to articulate it. So that's what we try to do, and many of my colleagues. And also what I specifically like to do is also provide a scientific basis, a formal description of what we say, and not just saying genes are not everything, right? So many people have said that. So now specifically on your question on cancer and non-genetic aspects, I believe in genes. I not belong to those who say genes don't matter at all. Some people believe that genes are just a storage to reconstitute the protein, and everything happens in protein level metabolism. And when a protein gets old and degraded, then you have a gene to remake that. That's not what I mean. Genes are important. They regulate each other, right? And when they're mutated into something else. So now the reason why we think that the most simple thing, since you ask a very complex question, why gene alone as an entity that we must think of changing in order to explain a change in phenotype, why that is sufficient is most prosaically manifest in the fact that a genome has many phenotypes, right? The same genome command the phenotype of every cell type. Identical twins are not the same, and so on. So something is going on, and that is because one gene doesn't map into one genotype, but in many phenotypes. That is essentially development. And therefore, we don't need to postulate an alteration of a gene to a new gene through mutation in order to explain a new phenotype, which is a pathology. So that's a core idea. And you mentioned, yeah, and with that you can explain non-genetic transition into malignant states and also reversion, because mutations, it would be very hard. People in the early days of bacterial genetics, they try to see if there are reverse mutations. Of course, it can happen by chance, very small probability you have to select for that. So I think the reversibility of phenotypes is just a fundamentally probably the strongest testament to the fact that we have a dynamics going on that is independent of mutations. Mm-hmm. Now, there are many details, why does it not happen more frequently? Why can we harness that potential for therapy? Well, those are different questions.
Sure. Well, let's go there next. And I just want to mention again for the audience, Dr. Wong just had a pretty seminal paper. It's entitled "The End of the Genetic Paradigm of Cancer," PLOS Biology. It was March 18th of this year with the Soto Sonnenschein team up at Tufts, sort of outlining everything we've just discussed for the last half hour. So we'll put a link to that paper in the show notes, because I think it's a nice encapsulation of the last couple decades of your work in this specific area. But I'd like to go now, Sui, sort of into we have an alternative theories here, of course, for how we look at cancer. Let's look at some of the interesting treatment strategies that you've developed concepts for over the years. And I sort of put these in different, I don't know, we'll create some classes here. We'll create something, we'll call it epigenetic landscape modifiers. We'll call attractor modifiers, whatever. We can brand them in different ways. But I think that the first of these, which is quite interesting to me, it has to do, again, looking at an epigenetic landscape, something you've written about in terms of ultimately, we have current treatments, whether it's chemo, radiation, and so forth, do an okay job of eliminating tumors, but cause all sorts of problems in the cellular microenvironment where the tumors are. This leads to, obviously, you've written about secondary tumor formations and all that, creating is something you call these tiny cytokine storms along the way. And sort of one of the solutions, one of the concepts you've created is that of, conditioning, I guess, the microenvironment, conditioning the landscape with in these resolving anti-inflammatory type compounds. I think this is just an interesting, again, a case where you're not dealing specifically with the tumor, but dealing with all the trouble that's been left behind by the stuff that's come first. Say a little bit about this concept. I think it's kind of interesting.
Yeah, thanks again for the summary. Yeah, so it's at the core is essentially the notion "thou shalt not kill the tumor cells," and that's counterintuitive because most people don't think of what happens if you kill tumor cells. Just drug companies they screen for maximally potent killing agents. Mm-hmm. But let me back up a bit because there are many paradigm shifts. And I know you mentioned that paper, which the title we didn't fully choose as the editor, and it's probably exaggerated, hyperbolic. Of course,
I liked it.
Genes are important. And by genetic paradigm, at the end, is meaning that we explain everything solely through the action of genes. So there have been paradigm changes. A big one that I was very honored to witness from close up when Judah Folkman proposed angiogenesis. Of course, there are always people before who have proposed that, but systematically promoted angiogenesis as a process in a tumor. That opened the field to, as you mentioned, the microenvironment. So the tumor is just not the cancer cells, very malignant, that grow, but there's a tumor microenvironment or stroma that is all the cells around the tumor, which are not normal inflammatory cells, fibroblasts, these support cells that become different without mutations, and then the blood vessel cells that then create new blood vessels that the tumor needs to grow because it needs nutrients. That's called tumor angiogenesis. So Judah Folkman opened the door to think beyond the actual malignant, so-called mutated cancer cells. And then that also opened the door to a lot of research that resurrected the whole idea of immune surveillance, which is the immune system, that how I start my career, can the immune system actually tumor at bay. So all these are so-called non-cancer processes, microenvironmental processes. And the idea to change that microenvironment instead of killing the tumor cells then arose in the form of anti-angiogenic therapy, so therapy directed against blood cells, and activating the immune system to activate those cells that recognize the tumor cells as something pathological and therefore can kill them. So that is, we need to know this as a backdrop. Now, I went back to focus on what happens when the chemo kills tumor cells. And that's part also of a new view in biology, and that is you don't have a cancer cell that you can draw as a cartoon and say, I'm going to block this and these essential biochemical steps in order to kill cancer cells. But you have millions or hundreds of millions of cancer cells in a tumor. If you now, as you know, if you do experiment, you increase the dose of a chemotherapy drug, then more and more cells die. That's a dose response curve. Right. So there's always, for any, however potent drug, some doses where 90% of cells die, but 10 don't because of the diversity of a population. That's how nature creates resilience, by having this intrinsic diversity or heterogeneity. So what happens to these 10% surviving cells that have seen the tumor, the drug, the anti-cancer drug, and survive? And now with single cell analysis, and so we learn that these cells are totally stressed out, right? Yeah. They're not normal, they're ready to resume and regrow, and then you cannot come back and double down to finish off. That's what people initially thought. Many still think. That's why you want to achieve so-called maximally tolerated dose, MTD, to kill as many cells as possible. But if you increase the dose, and there are always cells that survive at whatever dose, because you never reach the total highest dose for a complete annihilation of a tumor, then those surviving cells, the so-called non-killed cells, they don't have a resistance mutation in them. It's not selection like Darwin said, but they are plastic. We talk about cells being able to acquire many phenotypes. And it turns out that they activate a response that nature has pre-wired our genome to do, and that is, if you see toxic injury of your neighbors and of the tissue, you try to stimulate regeneration. You become like stem cells. You acquire stem cell properties. You acquire property to shut off the immune system, because regeneration should not interfere with the immune system. You stimulate the blood vessel growth. And so you are killing many cells. Yes, that's good, but you're injuring the others that become more malignant without mutations. And therefore, a cancer-killing therapy, as opposed to other type of therapy, is a double-edged sword. You want to eradicate as many cells as possible, but you want not to, anger the surviving cells. And sometimes it works. Sometimes there are so few surviving cells, but most of the time there are sufficient surviving cells, and they all prepare for regeneration. That's how cells are supposed to do. But in a tumorous tissue, this regeneration is pathological. So it overshoots. It cannot restore the normal tissue integrity, which is a feedback for any system to shut off regeneration. So that's why we have now the old notion by Virchow 100 years ago that the tumor is a wound that never heals, or a regeneration that never stops, a pathological, aberrant, out of control, runaway regeneration. And that's what we have. And with that come all the nasty properties, including the ability to shut off the immune system which tries to fix things and kill off pathogens and pathological states. And so at the end, it's a complicated optimization problem of a double-edged sword attack.
And staying in the sort of same sort of therapeutic class again of landscape modifiers related to this, and again this sort of thou send out kill theme. You just talked about maximal tolerated dose concepts there. One of the concepts related to that, which you've written about, is this principle of minimally effective dosing and adaptive therapies, and where sort of, as you say, the competitive release of these resistant cells could be slowed down or stopped if we just think again of how we might start with a certain dose, but we got to reduce it because then we can taper off what you were just explaining. We talked with Susan Domchek here at Penn, who's in charge of what's called a cancer interception program for the BRCA group. And it seems kind of similar there that, hey, this is not, in immuno-oncology, this is much earlier where, hey, if we could start with much lower levels of some of these therapeutics in a adaptive treatment protocol, we don't get to that, as you were saying, the mini cytokine storm events.
Yeah.
Say a couple words about that.
Yeah, that's a good point. Very important to bring it up. I like the adaptive therapy very much, but the initial idea was based on a different concept. And we do a lot of things based on so-called, insufficient explanations that still turn out to be appropriate. So this adaptive therapy concept and competitive release is still based on the old notion that cancer is due to mutations. You kill off a lot of cells, then not too much, and the other one, if you kill off too much, then the remaining cells are automatically resistant cells, and they would take over. So you don't want to avoid that. So you gently kill so that you don't trigger this competitive release of the more malignant cells, and then you titrate just the optimal dose, not to push too much the selection of resistant cells. But that idea is still based on the notion that cells are rigid. They don't have plasticity. They don't change their phenotype. They are just more resistant because of a mutation that change their blueprint. So they are different species, a different class of entities. And I think that theory of adaptive treatment has to be modified by embracing the fact that cells are plastic. They change their phenotype without selection because of the injury that they see. When you kill a lot of cells, all the dead cells, they send danger signals. I forgot to mention that earlier, again, to alert the tissue that there's an injury there, and you better become a stem cell and divide more. So it's a different idea underneath, but the overall idea of starting slow as opposed to maximally tolerated dose, the dogma for many years, is right on. But it works only in tumors because it's dangerous. It takes courage to not go in with full force. From the war on terrorism, and I write about that. When measured counter measures are important, and we have to learn that in cancer. But it works only if you can gauge how well you've been doing. So you need a biomarker for tumor shrinking and coming back. It works for some tumors like prostate. You have in the blood, you have markers, and others we can monitor by imaging. So for adaptive therapy, the challenge is to find a tool to monitor progress, so that you're doing the right thing. You need this back loop.
Yep. Yeah. Perfect. Let's transition now to another therapeutic class, which I'll call sort of attractor modifiers. And here I'd like to, it was interesting. You published, this is before the Yamanaka-Gurdon Nobel Prize. So the year before, in 2011, you published an interesting paper. It's entitled Understanding Gene Circuits, Cell-Phase Branch Points of Rational Cell Reprogramming. And you basically talk in this, so it's kind of easy, or not easy, but to switch a cell lineage. But ultimately, when you look at a body or an organism, you got 100 different cell types, planning itself without any specific instructions. reprogramming, dedifferentiating, redifferentiating, and so forth. I mentioned B. Mintz before, and sort of her work in the 70s on cancer reversion. We which itself was based on older work in the 40s and 50s on limb blastema reprogramming. Nature has really good ways to take cancer cells and turn them into normal tissue, and I think this is an area that holds a tremendous promise. I don't know if it's going to be based on Yamanaka factors or something more complex, let's say, per an epigenetic landscape, but love to get your perspective on sort of modifying attractor states via some of these principles that we're learning about.
Yes, very important. It's more theory because I don't think it will work. I can tell you why.
Please.
The two premises I need to introduce for you to understand my answer. First, the set of genes I mentioned create many configurations that are organized in many attractors. Some of them evolve to be harnessed by evolution to represent cell types. But mathematically, there are many more attractors that just due to the mathematics of 20,000 genes interacting with each other in some form of a gene regulatory network architecture. So only a subset is used. The others are potential states. And as I mentioned earlier, I view all pathologies as a system entering these adjacent potential states that are never realized and never fine-tuned by evolution because they were never exposed. And so an old idea, again, going back to Waddington and Kaufman especially, more explicitly, is that cancer is when cell accidentally enters these unoccupied attractors. That's the attractor theory of cancer. Now we can use this to explain mutations. Mutation essentially, if you look at many are in transcription factor, in chromatin modifier, right? Take 2 is one. The lower the energy barrier that separates the normal attractor of cell types and the cancer attractors. And evolution, if you look at the evolution of tumor suppressor and the evolution of developmental trajectories, has tried to prevent cells from being stuck in these alternative pathological, never evolved, and very faulty gene expression profiles that are just not physiological. So cancer is accidental entry into such an attractor state, either due to perturbations of gene expression profiles or due to mutations that lower the entry barrier. And that's why, because of that picture now that I hope you have in mind, you can, in principle, imaging a reversion. But unlike normal immature cells, which evolution of development, evo-devo, has carved very nice path of going to the next stage and then end up in the mature cell phenotype attractor. The cancer attractors, they're pathological because they have never been optimized by evolution, so they are not well drained. So it's very hard to get out of them. And we don't know how to do that. You mentioned Yamanaka, going back to an embryonic state. Again, in principle, you should be able to modify any complicated gene expression configurations and move from any state to any state. And I think we proposed that before Yamanaka, that in principle you could do that with a set of a handful of transcription factors, but we don't know how. And because now comes details, the quantitative details matters in biology, and that is the relative energy barrier height. They are not, it's a different issue. They're not true potential energies, but some sort of a effort energy to act against the constraints of gene regulatory network. Let's call it quasi-energy. So that height in terms of quasi-potential energy between attractor states is carefully wired is determined by the gene regulatory network, which is carefully wired by evolution to allow only certain transitions and make others very hard. And so there's clear evidence that some mutations just reduce the energy barrier such that you can easily slip into these alternative attractor states that are never evolved and never meant to be occupied and therefore not drained into normal states. That's why differentiation therapy or reversion is so hard. But it happens in some natural experiments. You mentioned also if you put the cancer cell into a normal tissue, and that's Soto and Sonnenschein that have promoted that in our piece, the so-called reprogramming or renormalization by putting a normal cell in an environment of normal tissue. That happens. It happens, I think, because the normal tissue provides so many, a gazillion of normal developmental signals. And so, again, a path through this high-dimensional space on the landscape means you change many, many genes in a very particular manner. And we just don't know how to do it in most cases, or we don't know how to do it efficiently. Again, I point to the importance of the actual quantitative numbers. The Yamanaka, experiments is very inefficient. 1 in 10,000 cells get reprogrammed. For cancer, you need to reprogram not 99% of the cell, but 100% of the cell. And that's why it doesn't work. Yes, you can see individual cells renormalized if you put the liver hepatoma cells back into normal liver, and so many normal signals surrounding this cancer cell would somehow reprogram it. But the signal is complex and the efficiency is very low. So to cure cancer, you just need to have 100% yield, and that's what we fail at.
No, it's again, it's I wanted to get your perspective on it, a very complex area, and what's I guess we're at the embryonic nature of it, but again, I just wanted to, as someone that's looked at this, get your insight. So I appreciate that. So, we talked a lot about cancer, but you do much more than that. And love to hear a little bit more about some of the other initiatives you have going on at ISB. And one of your most recent papers was quite interesting and had to do with Alzheimer's pathology and sort of looking at the different transcriptomes and how, again, cells in the brain, again, in sort of if we think of the landscape and the different attractors, potentially tilt towards these antigen presenting sort of T cell recruitment attractors and away from anti-inflammatory subtypes. What are you learning in Alzheimer's? Because clearly this is a big one that we know is a tidal wave is coming our way, and we don't have a lot of great therapeutic stuff going on. But
so I think you're referring to mostly the work driven by Andrew Baumgartner, a physicist. There are many aspects here. One is, there we look not at the neurons, but at the microglia, which is a white blood cell macrophage cell in the brain. But one notion there is that, again, you do have pre-existing states, and then you have transition states that are less, the hilltop between the attractors that destabilized. We can now, using single cell analysis, measure whether a cell is in such a destabilized intermediate state. Again, many people have postulated that these transitory states that are on a hilltop and therefore have many fade options in going multiple directions. I think we provide the mathematical foundation for that. So the question now is which states of the microglia or these macrophages in the brain states are good and which are bad. And that brings to a slightly different topic, which is also important in cancer, and that is, it's not about the cancer cells, but the microenvironment. And same thing with Alzheimer, right? These microglia cells turn out to be very important. And that relates to your point earlier when you ask about consequence of cancer therapy. So we don't think of what happens when you kill a cell. There's a dead cell body, and you create a lot of dead bodies. It's just not good. It creates inflammation. It has to be cleaned. And that we understand very little. Every time you have a lot of tissue injury, and that tissue injury, you probably know from textbooks, apoptosis is defined by cell death without causing inflammation. That's actually not true. It causes a lot of inflammation if there's enough cell death. Individual cells dying is fine. We couple up by these macrophages. If there's a lot of cell death, then you create a debris field that creates inflammation. And also inflammation is not good for Alzheimer, and it's not good for cancer. Especially now that we know, in the case of cancer, inflammation, while important for the immune system, it also shuts down the immune system. And that's why immunotherapy is such an uphill battle, because cancer is surrounded by a totally changed microenvironment that is very immunosuppressive. And so what we want is to gobble up dead cells, not cause a bad information that causes more injury. And that's true for Alzheimer and for cancer, right? And I think we have a drug now in development, hope to get into phase 2, developed by CITES that I'm advisor of, that does that. So take a broader view, reprogram the environment of your pathology, the microenvironment, tissue, as opposed to just the culprit cell, which is the neuron that produces this beta amyloid in the case of Alzheimer, and is dying, or cancer cells, the proliferating. You want to reprogram the stromal microenvironment. And again, many people try to do that, called stromal reprogramming. So a more encompassing, broader view. If I can focus on cancer, because there we have another dilemma. And most reason why most therapies don't work well is most interventions are double-edged swords. So for example, we kill cancer cells, create this debris, create inflammation, and all the stress response promotes the cancer. And I also mentioned inflammation inhibits the immune system. So then the question is, can we create minimal inflammation due to the cancer cells dying, but keep that at bay and steer the response towards activating the specific anti-tumor immunity, because the dying tumor cell release all these tumor-specific antigens. So we want to activate what is called the adaptive immune system, the T cells that specifically recognize cancer cells without creating too much damaging inflammation. Again, it's always a double-edged sword. We have to optimize carefully, and we think that our new class of drugs, resolvins, can do that. They stimulate macrophages to phagocytose or eat up dead cells without creating inflammation. But then, as we know, just fine, and I don't probably not publish it soon, they dramatically activate the so-called antigen presentation. So the macrophages, these white cells that eat up the tumor cells, will present the tumor antigens to the T cells. And only then can these checkpoint inhibitors actually work, because the antigens of the tumors to be presented to the immune system. So it's like a vaccination. Yeah. Now, the problem with the whole immunotherapy is, again, this dilemma that, like Alzheimer and cancer, these are tissue lesions that are sterile. They're not caused by bacteria or virus. And those sterile tissue lesions have a different effect on the immune system than those caused by bacteria. And we are still learning about those type of sterile information. How can you activate the immune system to clean up if there is no obvious pathogen?
Right.
So it's a tough problem.
No, but again, it's exciting the way you're thinking about it. So kudos to that. I very much appreciated that work, especially what we're dealing with let's just say biotech not doing a great job with Alzheimer's and dementia, but great. So one last thing I just want to ask about while we have you today, and I really do appreciate all your time and insight. In the recent Soto Sonnenschein paper, there's this paragraph entitled "Lessons from Organogenesis," and you're making the analysis and comparison between the stroma and how tumors potentially are guided or not, and organ formation, and this loops kind of full circle back to sort of where we started off in some of your work with Don Ingber and thinking about this principle of how cells ultimately sense and sort of this bridge the higher level needs of the structure they're forming. Any interesting stuff happening on the organogenesis, morphogenesis front in this era of regenerative medicine, where unfortunately we still are thinking primarily about cells, but the regenerative biology history shows us that it's cells, it's extracellular matrix, it's morphogenetic gradients, it's neurons, it's vascular genesis. Any exciting things happening in 2025 on that regenerative front that you could tell us about while we have you today?
Well, unfortunately, not exactly, because it's just hard, right?
Yeah.
regeneration, of course, inherently has a risk to create cancer.
Sure.
But since you mentioned organogenesis, I think the simple way to view it is cancer is a pathological organogenesis process that has not been tamed by evolution to create something useful. So we don't have this feedback loop that, okay, now you have to stop because you've created something useful.
Yeah.
And it is essentially, it has all the elements of organogenesis, but that is not orchestrated. And something that is so chaotic and broad benefits from this somatic selection, is mini evolution in your body, because the fittest will survive. And in regeneration, we try to prevent that. Everything has to be a loop. We need to stop when a desired tissue is created. And I don't know, that you will go beyond what we've been thinking about. Hope come from studying organogenesis in simple systems. I don't mind if it's a mouse or organ on a chip, on Ingber's work, or even just organoids, and figure out in a more formal framework how we can control it from a way, not bottom up. I think I made also a mistake, too much bottom up thinking, but we need a more top down thinking, right? How to manipulate, understanding the larger scale feedback loops that and can help you harness self-organization into some more meaningful tissues. And so in that sense, tumor is reverting back to a meaningful, regeneration.
Right. Fascinating area. Sui, I know that in a couple of days you have a research roundtable coming up that's going to be online. It's entitled "Cracks in the Cancer Paradigm." You'll be talking about the recent essay. Anything else coming up for 2025 that we should know about in terms of your work, other conferences, anything else that you want to mention while we have you today, please?
Yeah, we continue to work on details of what I've said. So what we have now, one result from very talented undergrad student, Andrew Taylor, is that we look at in detail the genes I mentioned that are upregulated in cells, in cancer cells, have survived cancer therapy within 2 days. So this is not selection and evolution. And so many genes that survive in cancer cell upregulate are exactly those that promote cancer or are markers of bad prognosis. That makes it very hard. So I think the upcoming team, now you mentioned the roundtable, which is to non-scientists, is that we all face the same type of problem when we try to intervene and steer a complex adaptive system with a punctual intervention. And that is, everything is double-edged sword and can backfire if you are not careful. So I try to, and I've reached out to politicians, right? They're much better, especially, I've mentioned that military leaders. Every intervention in war is double-edged sword. You cannot just kill all terrorists. That's like trying to kill all tumor cells, because the surviving one, you never manage to do that for all kinds of logistical reasons, and the surviving one will be angry at you, and they learn, and they regroup, and they are much more motivated. So everything's a double-edged sword, and a lot of strategic thinkers, in politics, in warfare, are much better at taking very measured, countermeasures against this uprising. And I think in biology, we need to learn to do that. We have so much to learn from other people trying to steer complex systems and to deal with them. And biology is just so primitive, just go and nuke the cancer cells. And now with smarter and smarter bombs, more and more specifically, it's not going to happen. And that's why we think there's a rethink that has taken place in many other parts of society, but not in molecular biology, which is very pedestrian in terms of, a more holistic thinking.
Absolutely. Well, I very much enjoyed this conversation. I have followed you, Sui, for years now in your work, and I look forward to continue to follow where you're going with all this, because it's truly transformational, paradigm shifting, whatever you want to put behind it, but just really impressive. Again, for everybody that is going to be listening to this episode of the show across the various podcast networks, or who will be watching on the YouTube channel, again, you've been spending time with Dr. Sui Huang, Professor, Institute for Systems Biology. Sui, I want to really thank you for taking the time out of your schedule to talk to us about what you're up to, for walking us through some of very difficult concepts. Obviously, thank you for everything you do in sort of the therapeutic space, new thinking. And as we like to say on our show, thanks for helping to create a better tomorrow via the development of these types of concepts. Really an awesome story, and I look forward to continuing to see what you do with it.
Yes, thank you very much. I really enjoyed your very cogent questions, and it's really a pleasure. Thank you.
Really great having you.